Hello everyone, my name is Sina, and today we'll be talking about the bus file. Now bus stands for Barcode UMI set, and a bus file is a form of reduced alignment data that usually encodes information from a standard single-cell RNA sequencing experiment. Let's take a look at an example bus file in standard text format. So I have an example here. Now you can see that there are multiple columns. Each column corresponds to an element of a bus file. The first column corresponds to the barcode, or the B in bus, which is a sequence of nucleotides corresponding to a sample or a cell. The second column corresponds to a UMI, or unique molecular identifier, and this is what tags individual molecules in a single-cell RNA-seq experiment. The third column corresponds to a set of transcripts. Now this set of transcripts stores alignment information about the read that generated this bus record. And the fourth column corresponds to the number of times that these three elements have been seen in the bus file. Now I just showed you an example of a bus file in text format, but we usually encode a bus file using a 2-bit encoding because this saves on space. So let's see how that 2-bit encoding is performed. So we have 4 nucleotides, A, C, G, and T. Each one of those nucleotides can be encoded using 2 bits. So A can be encoded using 00, C can be encoded using 01, G can be encoded using 10, and T can be encoded using 11. Now let's just go ahead and encode a short sequence of nucleotides using this binary encoding method. So let's say we had A, G, T, C. So the encoding for that would be A corresponds to 00, G corresponds to 10, T corresponds to 11, and C corresponds to 01. So as I mentioned before, the reason we use this 2-bit encoding is because it saves space on disk. So let's go ahead and take a look at an example bus file in binary format and its corresponding text format. So I have an example here. So in the terminal, we'll do ls-lht, and we see that we have the bus file in text format, which is 26 megabytes, and we have the bus file in binary format, and it's 24 megabytes. Now this is a small data set, but usually these files can get much, much larger, and by using the binary encoding, we can save a lot of space. So now let's go ahead and look at an example bus file in the binary encoding. So I have one right here, so if we try and open it up in VS Code, it'll give us a warning, and we'll just use our built-in text editor. And you'll see that it's just a bunch of characters that really mean nothing. You can see a little bit. There's bus file produced with Callisto, but because we haven't given the format instructions to VS Code, it doesn't really know how to read this file. But we can actually use bus tools, which is a set of command line tools, in order to view this file on the command line. So there are 2 other files associated with a bus file. The first one is a transcripts.txt, which is simply a list of all the transcripts that were used during the process of generating your bus file. The second file is what's called a matrix.ec, or an equivalence class matrix, which maps numbers corresponding to equivalence classes to sets of transcripts, or in this case, a single set of transcripts. So the first equivalence class, or I should say the 0th equivalence class, maps to the 0th transcript, and so on and so forth. Now, usually these are one-to-one, but the reason why we call these sets is because there can be sets of transcripts which share similar sequences, and as a result, they share an equivalence class based off of the equivalence relation where a subsequence of those transcripts are the same. So for example, this equivalence class is comprised of these transcripts. So there are 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 transcripts. So this means that these 10 transcripts share some subsequence within within the set of nucleotides that comprise that transcript, which make them which are the same. So if we're looking at a bus file, so if we go back here, this third column corresponding to the set now corresponds to a set of transcripts that the biological read associated with this record belongs to. I should also clarify that the numbers contained in the set for a single equivalence class, which correspond to individual transcripts, actually correspond to the row in the transcripts.txt file that I showed you earlier. So for example, for this equivalence class, there are 3 transcripts, and they each occur on row 4,974, 4,975, and 4,976. So if we wanted to find out what those transcripts were, we would go to the transcripts.txt file and find this exact row. But keep in mind that these are 0 indexed, and the numbering system on the left-hand side is 1 indexed in VS Code. So it can be a small gotcha. So if we go to find 4,974 in the transcripts.txt file, so this is 4,974 in the 1 indexed frame, but in the 0 indexed frame it would actually be this transcript here, because we would just have to add 1 since the counting would start at 0 instead of 1. So now if we want to view a bus file on the command line, we can actually use bus tools, which is a suite of tools that we recently published which allow you to manipulate bus files. And the way that you would do that is using the tool bus tools text. And if you just run bus tools text, you can see how you can run this. So usually the way I like to run it is by running bus tools text using the -p to pipe the output of the command to a file or to the terminal. Output.bus, so this is that binary encoded file we saw earlier, and then I can just pipe it to head so I can see the first 10 entries. And you can see these 10 entries correspond to the 10 entries that we saw in our text file here, and that's how we can view bus files. So I hope that was helpful. If you have any questions, please feel free to leave them in the comments below. Thanks.